Hello, Vishwanath. Good morning.
Hello. Good morning.
Can you hear me?
Hello.
Hello, hello. We'll be starting in just 2-3 minutes.
No problem. No issues.
Yeah. You have this thing from the department or from your home? Oh, department.
No. This is from home.
Home. Acha, acha.
Okay. Yeah.
And good night. त्याला इथलं दिसतं का त्याला फक्त आणि तो पण आता ऑफ केलं ते दोन आहे त्याचा रिमोट नाही का म्हटलं मग कॉम्प्युटर घेऊन आले म्हटलं इन केस लॉगिंग वगैरे करायचं इतक्या वेळ कोणाकडे चंद्रमुखी मॅडम हो सांगितलं
हेमांगे
हॅलो
हॅलो हेमांगे
सर बोला
व्हॉइस क्लिअर येतोय व्हिडिओ तुमचा ऑन नाही अजून हा ओके व्हिडिओ ऑन झालेला आहे
सर माय
यस यू आर ऑडिबल अँड युअर व्हिडिओ इज ऑन
ओके असंच बोलायला लागणार ना Good morning, everybody. I'm extremely happy to welcome Professor B.S. Vishwanath for this Viva and of Ms. Hemangee Damame. This is, for me, this is my last student's PhD Viva, so I'm very happy that Vishwanath is the external referee for this. I would just briefly like to introduce Professor B.S. Vishwanath. He's completed his PhD from Mysore University, Manas Gangotri Department, where I happened to work for some time. For 2 years, we were working together. Then thereon, he went on to Switzerland for his postdoc. For several years, he was working there. and then came back and joined the Department of Biochemistry, Mysore University as a leader. Then went on to become a professor and head. He has also served as the registrar of the Mysore University. His area of interest is on snake venoms. He has worked extensively on snake venoms and now on inflammation. His recent papers and even what he's worked in Switzerland is basically on inflammation. Done very excellent work, have a lot of publications to his credit. With a small introduction, I welcome Professor B.S. Vishwanath to this Viva. Can you hear me, Vishwanath?
Yeah. Thank you very much. Thank you very much for the kind words. Good.
Oh,
I
thank you
can
so
hear.
much.
Well, it's very good morning to all. Hello, sir.
She's
Hello.
I'm heading, yeah.
She's Professor Mrs. Jyoti Jadhav, who is heading the biochemistry department. She
That's
will
good.
be starting the Viva.
So today I welcome you all, Professor B.S. Vishwanath sir, Arvindekar madam, and all my colleagues and students from all the Department of Biochemistry, Biotechnology, Microbiology, and PhD as well as MS student. On behalf of Department of Biochemistry as a head, I welcome you all for the Viva Voice of Ms. Hemangee Damame, and as Madam said, she would be the last student of Madam. How many students you have, Madam, completed until now? Yeah. And this event we are celebrating because after a long gap, we again went together after several years, 2 years, and that's why first I welcome you all, and I request Hemangee to present her thesis. Thank you, sir.
Thank you, ma'am.
Physics just coming out.
That's fine. पूरी तरह से अपना प्रॉब्लम है जब नाम मत पटकन
Sir,
शेयर के लिए दिसता का विद्यास करना शेयर
recording.
More.
इथे. चला.
कुठे
She copies it, what I write, Hemangee.
हो.
मला एक दिन खाली खायला पाहिजे. शिवाजी विद्यापीठ संस्थाचा शिवाजी
हो मॅम.
Recording ते होस्ट पण आहे
होस्ट शिवाजी.
तो बरोबर अजून बोला
Good morning, all of you. I am Hemangee Damame, working as PhD student under the guidance of Professor Arvindekar ma'am in Department of Biochemistry. Today, I am defending my viva on research topic entitled as Study of Herbal Formulation in Treatment of Type 1 Diabetes mellitus.
Vishwanath, can you hear it?
Yeah. I can hear. No issues.
Yes.
Let's start with diabetes mellitus. Diabetes mellitus is chronic metabolic disorder of glucose metabolism, which results into hyperglycemia. And this is due to the lack of insulin, defect in insulin signaling pathway. And currently, diabetes mellitus is rising from 4.7% to 8.5% in adult population. Diabetes mellitus is classified into three major types, that is type 1 diabetes mellitus, type 2 diabetes mellitus, and gestational diabetes mellitus. So let's start with type 2 diabetes mellitus, which is caused due to insulin resistance, in which target cells fails to respond to insulin, which is secreted by insulin producing beta cells. About 95% of world population affected with this type 2 diabetes mellitus, and this is due to the insulin resistance. Hence, it is necessary to control diabetes mellitus. So, our laboratory has been working in area of diabetes mellitus and secondary complications associated with diabetes mellitus, and for over 18 years. And our laboratory has been reported many herbal components having insulin secretagogue activity, alpha glucosidase inhibitory activity, insulin sensitizing activity, and many more. Let's start with chapter first, introduction and review literature. Now, the major type of diabetes mellitus, we focus on type 1 diabetes mellitus, which is organ-specific autoimmune disease caused due to destruction of insulin-producing beta cells. About 5 to 10% of all cases of diabetes affected in younger individuals below the age 20. And this type 1 diabetes is caused due to destruction of insulin-producing beta cells, which is due to the autoimmune attack. And different types of immune cells are involved in this autoimmune attack, that is B cells, T cells, CD4 T cells, CD8 T cells, imbalance of regulatory T cell and effector T cell, Th1 and Th2 imbalance, dendritic cells, and macrophages. Now, the actual mechanism involved in type 1 diabetes mellitus is shown in figure. So, destruction of beta cells is result of viral infections, dietary factors, and physiological stress, which causes the activation of dendritic cells. And this activation of dendritic cells, the taken of beta cell antigen alter the responsiveness of these beta cells. And this interferon secreted by beta cells which further activate dendritic cells, which stimulate CD4 T cells and CD8 T cells. So these CD4 T cells causes destruction of beta cells through macrophages mediated cytokines and activated antigen presenting cells. So, on other hand, the CD8 T cells causes the destruction of beta cells through granazine, perforin, and fast-fascial ligand pathway. So, this is the overall mechanism involved in type 1 diabetes mellitus. Now, the complication. As the type 2 diabetes type 1 diabetes has also causes the secondary complications, that is short-term complication and long-term complication. Short-term complications include hypoglycemia, hyperglycemia, whereas long-term complications include microvascular disease and macrovascular disease. Now, the treatment and prevention trial for type 1 diabetes mellitus is insulin therapy is the mainly treatment available nowadays for the type 1 diabetes mellitus. And different types of insulins are available in market, that is Lispro, Actapid, Humulin, Lantus, and Mixtard. In order to reduce the rate of mortality due to type 1 diabetes mellitus, many investigations have been conducted by researchers to search alternative therapy for the insulin. So, the primary trials were carried out by using diet free of cow's milk and gluten, whereas secondary trials were carried out by using the strategies like regeneration of pancreatic beta cell, transplantation therapy, and monoclonal antibody. But none of these have to be effective against type 1 diabetes mellitus. Now, research problem. Type 1 diabetes mellitus is caused due to destruction of beta cells, which further results into insulin deficiency. And as this type 1 diabetes occurs in formative years in small children, it impacts their childhood. Children are prohibited from taking sweets and taking the intensive sport activities and other barriers that impact their development. So currently, injectable insulin is a major therapy for the treatment of type 1 diabetes mellitus, which is painful, cost prohibitive, leads to immune response and other problems. Hence, there is need to develop an alternative oral therapy that is easily available, cost effective, and without toxicity. It is therefore attempted to prevent apoptosis of beta cells, regeneration of new beta cells, use insulin mimetics to lower blood glucose level by using herbal components. Now, the objectives of my study are develop a type 1 like diabetic animal model through repeated streptozocin injections and evaluating its diabetic status. Study of plant extract that can prevent apoptosis of beta cells through cell culture studies on mean-6 pancreatic beta cell line through the use of cytokines such as TNF-alpha, interleukin-1 beta, and interferon gamma. The evaluation of plant extract that can promote regeneration of functional beta cells from pancreatic ductal cell line PANC1 through beta cell specific staining, insulin production, and gene expression studies. In vivo studies in type 1 experimental animals by administration of combinatorial formulation of antibiotic, regeneration stimulating, and insulin mimetic plant extract and studying the serum parameters and histopathology of pancreas. So let's start with chapter 2, material methods. The study of antibiotic and beta cell protective activity of herbal plants in mean-6 pancreatic beta cells was carried out by using the experiment Adexin 5 and FITC propydium iodide staining, MTT assay to study cytotoxicity, nitrate measurement by grease reagent, ROS measurement by DCFDA dye, mitochondrial membrane potential measurement by rhodamine-1 dye, gene expression study using genes of caspase-3 and insulin secretion assay using ELISA kit, and the in vitro differentiation of human pancreatic ductal derived PANC1 cells into beta cell phenotype was carried out by using the experiment in vitro induction of differentiation of PANC1 cell line into beta cells, their morphological analysis by phase contrast microscopy, Dye-Thousand staining and Gomori staining, molecular characterization by mRNA expression study, functional characterization by glucose-stimulated insulin secretion assay, and the in vivo evaluation of combinatorial formulation having antibiotic, regenerative, and insulin mimetic action in experimental treatment of type 1 like diabetes in experimental animals was carried out with in vitro assays and in vivo assays, that is induction of diabetes, determination of blood serum, urine and lipid parameters, assessment of antioxidant enzyme system, glycogen content of liver, assessment of tissue fluorescence, histopathological studies of tissue pancreas, and measurement of insulin secretion in plasma. And all the statistical analysis was carried out by Grapher, Prism, and MS Excel software. Let's start with chapter 3, that is study of antibiotic and beta cell protective activity of herbal plants in mean-6 pancreatic beta cells. So, development of therapy option for the treatment of type 1 diabetes mellitus is hampered due to non-availability of experimental models. So, we are interested to develop a type 1 experimental type 1 in vitro model. So, firstly, this with the development of experimental type 1 model and further carry out the screening of herbal plants for the beta cell protective activity. So, for this study, we use murine pancreatic mean-6 beta cell line, which is derived from mus musculus and epithelium in morphology and cultured under the DMEM medium with 10% FBS under the standard condition at 5% CO2 and 37 degrees Celsius in CO2 incubator. These are the pancreatic mean-6 viable cells. So, let's start with part first, induction of type 1 diabetes using cytokines. So, it is found that beta cell loss in islet of pancreas was due to the products of cytotoxic T cells and macrophages, mainly pro-inflammatory cytokines, that is interleukin-1 beta, TNF-alpha, and interferon gamma. So, for this study, we selected these 3 cytokines, and initially we optimized the concentration for the apoptosis in mean-6 beta cell line by using apoptosis assay. So, let's start with interleukin-1 beta. Mean-6 cells were treated with gradient of interleukin-1 beta concentration ranging from 25 to 200 nanogram per ml. And after the treatment, cells were stained with Adexin 5 and propydium iodide. And cells then further acquired by flow cytometer and analyzed by Flowjo software. So cells are categorized into early apoptosis, late apoptosis, and dead cells on the basis of principle annexing 5 to negatively charged phospholipid present phosphatidylserine present on the inner limit of plasma membrane and DNA intercalating properties of propydium iodide. So, on the basis of this principle, cells are categorized into early apoptosis, late apoptosis, and dead cells. So, among this concentration, 100 nanogram per ml shows higher and significant apoptosis induction with early apoptosis that is 5%, late apoptosis that is 1.49%, dead cells that is 20%. So, this concentration was selected for the induction of apoptosis in V6 pancreatic beta cells. The graphically shown in figure as flow cytometer analysis of apoptosis induced by Karlov's treatment of interleukin-1 beta. So these are the 4 quadrant images of flow cytometer. The first graph shows the control cells, and B, C, D, and E shows the cells graph for the interleukin-1 beta treated cells. So 100 the graph D we discuss with detail in graph B. So the first quadrant is of live cells, and second quadrant is of early apoptosis, early apoptotic cells, which is positive for annexin 5 but negative for propydium iodide. Then third quadrant is of positive for the annexin 5 also, and positive for propydium iodide also. And fourth quadrant, that is double positive for propydium iodide. So on the basis of this principle, cells are categorized into these apoptotic events. So the 100 nanogram per ml concentration of interleukin-1 beta selected for further study. As same like interleukin-1 beta, we also carried out apoptosis assay by using tumor necrosis factor using the concentration 5 to 50 nanogram per ml. And the 50 nanogram per ml shows the high and significant and stable apoptosis induction than other concentration. And these are showing here the graphically representation which demonstrate V6 beta cells undergoing apoptosis when they are exposed to TNF alpha. So the concentration 50 nanogram per ml was selected for further study. Same as interleukin-1 beta and TNF alpha, we carried out apoptosis assay for the interferon gamma. In these V6 cells treated with different concentration of interferon gamma, that is 25 to 200 nanogram per ml. Among these all concentration, 50 nanogram per ml shows the high apoptosis induction and significant apoptosis induction than other concentration. These are images showing the effect of interferon gamma on V6 cells. That is 4 quadrant images of flow cytometer. So these are the total graph showing the total apoptosis by interleukin-1 beta, TNF alpha, and interferon gamma. And optimizer concentration 100 nanogram per ml, TNF alpha 50 nanogram per ml, and interferon gamma 50 nanogram per ml. So from this graph, we come to know that at this optimizer concentration, the total apoptosis is increased as compared to control cells. So these 3 dose concentration for respective cytokines are selected for further study. From literature survey, we come to know that combination of interleukin-1 beta, TNF alpha, and interferon gamma stimulated high apoptosis induction than individual cytokines. So we use these 3 cytokines in combination. That is cytokine cocktail 1 having the interleukin-1 beta 100 nanogram per ml, TNF alpha 50 nanogram per ml, interferon gamma 50 nanogram per ml. While cytokine 2 is interleukin-1 beta 50 nanogram per ml, TNF alpha 25 nanogram per ml, and interferon gamma 25 nanogram per ml. So
go back to that. So here you have 150.
Yeah.
Cocktail one you have this,
in cocktail same and cocktail 2 मध्ये. So among these,
whether it works with lesser concentration, it works.
Yeah.
so among these concentration, cytokine cocktail 2 having the concentration interleukin-1 beta 50 nanogram per ml, TNF alpha and interferon gamma 25 nanogram per ml shows the high induction as in figure 8C, figure 9C, and figure 10C. So from this, we can come to know that cytokine cocktail 2 induce higher apoptosis than cytokine cocktail 1. And this cytokine cocktail 2 is checked for 3 different time intervals, that is 3 hours, 6 hours, and 12 hours. So among these 3 time intervals, 6 hours treatment shows the higher apoptosis than 3 hours and 12 hours. So the optimum condition that is cytokine cocktail 2 for 6 hours was selected for apoptosis induction in mean 6 pancreatic beta cell line. For further study, we use camptothecin as a positive control. So we carried out assessment of in vitro induction of apoptosis by positive control, that is camptothecin, by using the concentration of 1 micromolar, 2 micromolar, and 4 micromolar. So among these concentration, 2 micromolar shows the higher total apoptosis than other concentration, that is 1 and 4. So these 2 micromolar concentration was selected for the induction of apoptosis induction, and CPT used as a positive control. So the figure 14 shows a schematic representation of beta cell apoptosis induction by cytokine cocktail. So it's established here about type 1 experimental models by using the combination of these 3 cytokines, which causes apoptosis induction and also enhances the reactive oxygen species levels, nitrogen oxygen species level, which leads to oxidative damage and also causes the decrease in mitochondrial membrane potential, and also lastly activate the caspase cascade activation, which further leads to beta cell death as in type 1 diabetes model. So we establish a pancreatic mean 6 cells as an in vitro type 1 model by using mean 6 beta cell. So the study is published in Journal of Pharmacological and Toxicological Methods, entitled as In Vitro Model Using Cytokine Cocktail to Evaluate Apoptosis in Mean 6 Pancreatic Beta Cell. Now let's part second, screening of herbal plants having anti-apoptotic and beta cell protective activity. For this experiment, we selected the plants, that is Fuchsia cerata, Justicia adatoda, Musea ferrea, Piper longum, Tinospora cordifolia, Umbilica officinalis, Terminalia jegula, Jasminum multiflora, Curcuma longa, Piper magnum, Eclipta prostata. These 10 herbal plants were selected for the beta cell protective action on the basis of literature survey and discussion with local renowned Ayurvedic doctors. So we carried out apoptosis assay by using these 10 herbal plants, in which mean 6 cells were treated with aqueous extract of these herbal plants for 1 hour, and then exposed these pre-treated cells to the cytokine cocktail for 6 hours. And after that, we carried out the staining of anethylene 5 and clopidogrel iodide by using apoptosis kit and analyzed the cells by using flow cytometer. So and percentage of apoptotic apoptotic events are categorized in table. So from this table, we can say that curcuma longa and Piper magnum show decreasing apoptosis in early phase, late phase, and also dead cells. So these curcuma longa and Piper magnum in 2:1 ratio was selected for the beta cell protective activity, and which is inhibited the apoptosis as compared to the cytokine cocktail 2. These are the flow cytometric images, shows the recovery of cytokine induced apoptosis using herbal extract. So control A graph is of control where apoptotic events are less as compared to, and B is cytokine cocktail treated cells where the apoptotic events are high. While the graph K shows the curcuma longa and Piper magnum treated cells, which shows the less apoptotic events as compared to cytokine cocktail 2. So this curcuma longa and Piper magnum was selected for the beta cell protective activity. So to check if this curcuma longa and Piper magnum has any cytotoxicity in mean 6 cells, we carried out assessment of cytotoxicity study by using the MTT assay. For this experiment, we use the concentration, that is 1 microgram, 2, 4, 8, and 16 microgram. So among these 5 concentration, the 1 microgram concentration per ml shows the high maximum cell viability. So this concentration is we selected as an effective concentration, and it is used for the beta cell protective activity. Then we carried out assessment of effect of curcuma-Longan papanogram on cytokine cocktail induced apoptosis. So these are the representative bright field images of V6 cells undergoing apoptosis. The first one is control cells, which shows the viable cells. So when the cells are treated with camptothecin, there is destruction of beta cells. And as also same in a cytokine cocktail 2, there is destruction of beta cells. While when we treated with these cells with curcuma-Longan papanogram, they show ability near about the control cells. So we can say that curcuma-Longan papanogram shows the beta cell protective activity against cytokine cocktail induced apoptosis. And this is diagrammatically shown in figure in figure 19. That is, A is of control cells, B is cytokine camptothecin treated cells, while C is the graph showing the cytokine cocktail treated cells, while the curcuma-Longan papanogram treated cell shows the less apoptosis event. That is, maybe they say there is decrease in apoptosis event. So the graph 20 shows the prokaryotic role of curcuma-Longan and papanogram extract. In this graph, it is come to know that cytokine cocktail shows the high apoptosis induction, that is total apoptosis, while the curcuma-Longan papanogram shows the decrease in total apoptosis. So then we carried out effect of curcuma-Longan papanogram on nitrate synthesis. So nitric oxide is one of the mediator of reactive nitrogen species, which causes the oxidative stress. So we measure this nitric oxide production by using Rees reagent. So the figure from this figure we come to know that nitric oxide production is increased in cytokine cocktail treated cells, whereas curcuma-Longan papanogram shows the rescued these cells from the oxidative stress. That is, there is the reduction of nitric oxide production. Then we carried out effect of curcuma-Longan papanogram on oxidative damage. Change in redox environment in cell is the one of the cause of cell loss and cell dysfunction, and this is characterized by ROS level. So after the treatment, we strained the cells with DCF-DA dye and acquired by the flow cytometer and analyzed by the flow cytometer. So after analysis, we come to know that the ROS levels are increasing in cytokine cocktail treated cells, whereas it is decreased in when the cells are pre-treated with curcuma-Longan papanogram. Then we carried out assessment of effect of curcuma-Longan papanogram extract on mitochondrial membrane potential, which is studied by using the rhodamine 123 dye. And the mitochondrial membrane potential is measured on the basis of retention of rhodamine 123 dye into cells and uniformity of mitochondria. So figure 25 shows the percentage distribution of mitochondrial membrane potential. So in the case of cytokine cocktail treated cells, the mitochondrial membrane potential is decreased. While when we consider the curcuma-Longan papanogram, the mitochondrial membrane potential is restored. So curcuma-Longan papanogram have the beta cell protective activity against cytokine cocktail. Further we carried out the gene expression study by using caspase-3, which is apoptotic marker, and insulin gene. So from this figure 26 shows the effect of curcuma-Longan papanogram extract on gene expression of caspase-3. In this figure, the caspase expression is increasing in cytokine cocktail 2, while the curcuma-Longan papanogram shows the decrease in downregulation in caspase-3 gene expression by threefold, whereas figure 27 shows the increase in insulin gene expression in curcuma-Longan papanogram treated cells, while it get downregulated in cytokine cocktail treated cells. So insulin genes are upregulated in case of curcuma-Longan papanogram treated cells. Then we carried out the evaluation of effect of curcuma-Longan extract on insulin secretion. We have interest to check if this curcuma-Longan papanogram have any effect on insulin secretion. So we carried out the insulin secretion assay by using insulin ELISA kit. So after that, from this graph, we come to know that curcuma-Longan Piper diagram increases the insulin secretion as compared to cytokine cocktail treated cells. Then we carried out antioxidant potential of curcuma-Longan Piper diagram extract by FRAP assay. So from this table, we come to know that as the concentration of curcuma-Longan Piper diagram aqueous extract is increased, the antioxidant potential is also increased. So curcuma-Longan Piper diagram shows dose-dependent antioxidant potential. So the figure 29 shows the schematic representation of probable mechanism of beta cell productive activity of curcuma-Longan Piper diagram herbal drug against cytokine cocktail. So from this figure, we come to that curcuma-Longan Piper diagram shows the protective activity against oxidative damage, decrease in mitochondrial membrane potential, and decreases caspase 3 expression, which is the three markers and which is the three stages at which our drug shows the beta cell protective activity. The study is published in journal Phytomedicine Plus, entitled as Beta Cell Protective Effect of Curcuma-Longan Piper diagram in Cytokine Cocktail Inducer Apoptosis in mean-6 pancreatic beta cell. So the conclusion of this chapter is interleukin-1 beta, TNF-alpha, interferon gamma at respective concentration 50 nanogram per ml, 25 nanogram per ml, 25 nanogram per ml for 6 hours successfully induced a stable apoptosis in mean-6 beta cells. Curcuma-Longan Piper diagram at 1 microgram per ml prevented apoptosis of beta cells. Curcuma-Longan and Piper diagram in ratio 2:1 showed no cytotoxicity in mean-6 beta cell. Curcuma-Longan and Piper diagram also exhibited antioxidant potential in concentration-dependent manner. Therefore, prevention of apoptosis can be a major therapy option in treatment of type 1 diabetes mellitus. So let's start with chapter 4, in vitro differentiation of human pancreatic duct derived PANC1 cells into beta cell phenotype using Tinospora cordifolia. So in vitro differentiation, for this experiment, we use the human pancreatic PANC1 ductal cell line. So type 1 diabetes is caused due to destruction of beta cells. So there is loss of beta cells. So regeneration of new beta cells from endocrine tissue is the one option. So we tried the in vitro, maybe one option for the compensate these beta cells. So we tried the in vitro differentiation of PANC1 ductal cell line into functional beta cell line. So for this we use the PANC1 ductal cell line, and these cells are cultured in culture condition, that is DMEM medium with 10% FBS under the standard condition, that is 5% CO2 and 37 degrees Celsius in CO2 incubator. And we use the Tinospora cordifolia as an inducer on the basis of study carried out by Dr. Ashok Saugule sir, in which the diabetic rat are reverted back to the normal with Tinospora cordifolia. So the overall protocol for the in vitro induction of differentiation by Tinospora cordifolia extract was carried out for 6 days, step by step. In this experiment, 1 into 10⁵ cells are incubated or supplemented with differentiation medium containing DMAM medium, ham cell, F12 medium, ITS solution, and Tinospora cordifolia. So insulin present in ITS solution causes the uptake of glucose, synthesis of amino acids and proteins, lipogenesis, and intracellular transport. Whereas transferrin present in ITS solution causes the decrease in oxidative stress during the regeneration. While and it is also act as iron carrier, while the selenium present in ITS mixture is act as antioxidant, and also it is cofactor for glutathione antioxidant system. And Tinospora is act as inducer for this differentiation process. So during differentiation, cells form the zone of activation. On the 4th day, cells shows the cells are aggregated to each other and shows the islet-like structure. And after completion of the 6 days experiment, the cells shows the actual islet-like structure. And they are observed with the help of bright field microscopy as well as phase contrast microscopy. The black arrows denoted here show the islet cluster formation. So we can say that there may be development of beta cell-like structure. So this is confirmed through the morphological characterization of these differentiated cells by using dihydrogen staining, which is exclusively used for the islet staining. So after this regeneration experiment, Tinospora industrial regeneration experiment, we carried out dihydrogen staining of this converted cell. So after staining, this cells shows the brownish colored color. So and after then also we carried out Gomori staining. It is used to differentiate beta cells from alpha cells. So after Gomori staining, the converted cells shows the purple color granulation. So from these two experiments, we come to know that there is development of beta cells from pancreatic ductal cell line. Sorry, regeneration of beta cells from pancreatic ductal cell line. So it is confirmed with molecular characterization of beta cells using mRNA expression study. So this experiment is carried out by using the genes that is insulin and PDX1 and which is characteristic marker for beta cells, and carbonic anhydrase, which is the marker for pancreatic ductal cells. So insulin and PDX level is increased in converted beta cells, converted cells by 2.8 fold and 2.2 fold. Whereas pancreatic ductal cells shows the carbonic anhydrase, which is get down regulated by 1.3 fold. So from this experiment, we can say that Tinospora has potential to induce in vitro differentiation of PANC1 cells into beta cells. So we have interest to check if these converted cells are functionally active or not. So we carried out functional characterization of these differentiated beta cells using insulin secretion assay. So for this experiment, we use 10 millimolar arginine as an enhancer for the insulin secretion assay, and 5 millimolar and 25 millimolar glucose as a basal concentration, and 20 millimolar glucose as a high concentration. So these converted cells shows the insulin secretion from basal concentration to high concentration. So these converted cells are functionally active. We also check the antioxidant potential of this Tinospora cordifolia. So from this table, we also come to know that as the concentration of Tinospora is increases, the antioxidant potential is also increasing. That means the Tinospora has the dose-dependent antioxidant activity. So this is the schematic representation of regeneration of PANC1 pancreatic ductal cells into insulin secreting beta cells. So from this diagram, we can say that Tinospora cordifolia has potential to regenerate and help the differentiation of PANC1 ductal cell line into functional beta cells. The conclusion of the chapters are Tinospora cordifolia demonstrate in vitro differentiation of non-endocrine, that is pancreatic ductal PANC1 cells into beta cells. Differentiated cells showed the expression of PDX1 and insulin, that is the characteristic of pancreatic beta cells, and downregulation of carbonic anhydrase 9. The converted cells are functionally active in response to glucose challenge. Therefore, the plant components such as Tinospora cordifolia can induce differentiation of pancreatic ductal cells into functional beta cells, thereby increasing the beta cell mass. And the work is under communication in the journal In Vitro Cellular and Developmental Biology Animal. The chapter 4, which deals with in vivo evolution of combinatorial formulation in treatment of type 1 like diabetes in experimental animals. In earlier two chapters, we inhibited the apoptosis in main 6 beta cells and regenerated the functional beta cells from PANC1 ductal cells. So it is decided if this therapy can work in vivo situation, but however, it is not possible to get type 1 genetic model. So we use the induce the type 1 diabetes using ceftriaxone, and which destroy the whole pancreatic beta cells. And but in as we know that restoration of beta cells takes some time, but in the meantime, the high blood glucose is the fatal to the animal. So it is necessary to include insulin in the formation. So we include insulin mimetic, that is cinnamomum plant as an insulin mimetic in this formulation, along with Chirospora cordifolia and Curcumin long and Piper. And the plant Cinnamomum tamala is selected on the basis of study done by Dr. Tanaji sir. So, firstly, we optimize the dose of Cinnamomum tamala for insulin mimetic activity. We carried out the experiment, that is first in vitro study of glucose uptake, in which different concentration of distillate, 10% distillate of Cinnamomum tamala was used as a test component, and insulin used as a positive control. So the glucose uptake is increased at the 0.5 ml distillate concentration in both hemidiaphragm as well as soleus muscle. Same as glucose uptake, glycogen synthesis is also increased at the concentration 0.5 ml and in both as hemidiaphragm as well as soleus muscle. Then we carried out in vitro study of pyruvate uptake. The distillate concentration at 5.5 ml of Cinnamomum tamala shows the restricted the pyruvate uptake as like insulin. And also in figure 44 shows the effect of Cinnamomum tamala on glucose release. Here the glucose release is also emitted when the animal treated with distillate of 0.5 ml. So from this experiment, we can say that Cinnamomum tamala acts as insulin mimetic. We carried out assessment of antioxidant potential of Cinnamomum tamala. This is also from this table we also come to know that the Cinnamomum tamala shows the concentration dependent antioxidant potential. So then we carried out then we did with part 2, that is in vivo experimental animal design, in which normal male vista rats were divided into 4 groups as group 1, that is control rat, group 2, that is type 1 diabetic control, which type 1 diabetes was induced with cetrodrosin injections at a dose 55 mg per kg body weight and it is used twice. So after 14 days of injection, blood glucose level was checked for all experimental rats, and the rats which shows the blood glucose level above 400 mg per dl was selected as type 1 diabetic rat. So group 3 is insulin treated type 1 diabetic rat, and group 4 is herbal formulation treated diabetic rat, type 1 diabetic rat. So all doses were given twice a day for 30 days, a day of treatment. The treatment is restricted to 30 days as the diabetic control rat could not survive beyond the 30 days. So after 30 days of treatment, the animals were sacrificed and subjected by cervical dislocation and subjected for biochemical and histopathological analysis. So first one, in effect of combinatorial treatment on body weight, serum, blood and liver function parameters. In case of fasting blood glucose level and glycated hemoglobin, type 1 diabetic rat shows the high blood glucose as well as glycated HB, whereas the herbal formulation treated rat shows the decrease in glucose level as also and also glycated HB. So in case of liver profile levels are increased in case of type 1 diabetic rat, while our herbal formulation treatment shows the improvement in this lipid profile. And the liver function test, that is SGOT and SGPT, are increased in type 1 diabetic rat, shows liver dysfunction, whereas our herbal formulation treated rat shows the decrease in these enzymes. So then we carried out effect of combinatorial treatment on urine parameters. For this experimental hours of urine are used and the parameters like glucose and albumin are well resolved when the type 1 diabetic rat treated with herbal formulation. Then we carried out tissue fluorescence. Then we checked tissue fluorescence. So for this experiment, we select 5 tissues, that is liver, kidney, heart, lungs, muscles, and lens. So the total tissue fluorescence and specific fluorescence are increased in type 1 diabetic rat, whereas the tissue fluorescence decreased in case of herbal formulation treated rats, as like insulin. Then we carried out oral glucose tolerance test. So from this test, we come to know that insulin as well as herbal formulation treated rat shows the glucose lowering effect. While combinatory effect of treatment on liver glycogen content, in this case, the glycogen content is increased in when the rats are fed with herbal formulation. Oxidative stress is one of the key factors involved in the establishment of diabetes mellitus. So we want to check the antioxidant status of animal rats, experimental rats. So we carried out the superoxide dismutase experiment. So for this we take liver and kidney and we check the SOD levels. So SOD levels are decreased in case of type 1 diabetic rat, whereas our herbal formulation treated rat shows increasing SOD levels both in liver as well as kidney. Same as SOD, catalase also perform. From this graph, we can say that catalase levels are decreased in case of type 1 diabetic rat, whereas it gets increased in case of herbal formulation treated rat. And then we carried out third experiment, that is thiobarbituric acid reactive substance, that is TBRS, which is great increased into type 1 diabetic rat, whereas our herbal formulation treated rat shows decrease in TBRS level both in liver as well as kidney. So from these three experiments, we can say that the herbal formulation has antioxidant response against tetrodotoxin. Then we carried out histopathological assessment of pancreas from experimental animals. So for this we carried out the hematosaline use in histopathology study. So from this figure we come to know that the cellular and cellular architecture of islet is disrupted in type 1 diabetic rat, and there are also the presence of intracellular space in the beta cells. So while the insulin treated and our herbal treated rat shows the decrease in intracellular space, or maybe there is a restoration of beta cells. So we want to check if this pancreatic tissue is functional or not. So we carried out insulin secretion assay and we checked the insulin from the blood from each experimental animals by using the insulin ELISA kit. So in that case, type 1 diabetic rat shows the decrease in insulin levels, whereas our herbal formulation treated rat shows the increase in insulin levels. So the conclusion of this chapter is in vivo treatment with insulin mimetic Cinnamomum tamala along with Curcumin and Piper nigrum and Tinospora cordifolia showed improvement in serum parameters, blood glucose, and significantly demonstrated insulin production in treated animals. Therefore, the above findings demonstrate that type 1 diabetes can be treated by oral herbal components that prevent the pain of multiple insulin injections, cost effective and non-toxic on prolonged use. So the significance of my work is in an in vitro model for inducing apoptosis with cytokine cocktail of interleukin 1 beta at 50 nanogram per ml, TNF alpha 25 nanogram per ml, interferon gamma 25 nanogram per ml in mean 6 beta cells was established, and it can serve as a primary screening model for anti-apoptotic drug for type 1 diabetes. Second, the curcumin longan Piper nigrum in ratio 2:1 at 1 microgram per ml showed protective, anti-apoptotic, and antioxidant response against cytokine cocktail induced injury in mitochondrial mediated intrinsic pathway. And the third one is Tinospora cordifolia extract has potential to act as inducer to initiate the differentiation of PANC1 ductal cells into functional insulin producing beta cells. The present study is the one of the kind that attempts to prevent beta cell from autoimmune destruction of beta cells, regenerate the new beta cells from pancreatic ductal cells, and also use an insulin mimetic to combat hyperglycemia. This is the first attempt at development of combination of herbal extract of Cinnamomum tamala, Tinospora cordifolia, and Curcumin longan Piper nigrum as oral therapy option for the treatment of type 1 diabetes that can slowly replace the insulin therapy and can prove to be boon for type 1 diabetes. And therefore, the combinatorial therapy holds the promise for the treatment of type 1 diabetes. The future perspective of my study is, as the work is preliminary on human cell line and later through in vivo trials on cetirizine-induced type 1 diabetic rats, it has huge potential for the treatment of human patients and systematic study of mechanism, identification of precise bioactives, and conduction of clinical trials. These are the publications. The manuscript under communication and under preparation, the academic conferences attended during these years, and firstly, I thankful to Department of Biochemistry and the Animal Ethical Committee for providing lab spaces and lab facilities and workspace. I also thankful to Dr. Chandamukhi Garad ma'am for her constant help and suggestions about the Ayurvedic formulations of drugs and introducing me into the exciting world of Ayurvedic research. And I also acknowledge to Rusa Sarthi and Pers Phase 2 to give me a financial assistance during these years. And I also acknowledge RGSTC and Rusa for providing funding for our research. And I also acknowledge the National Center for Cell Science for providing all the cell lines. And I also acknowledge the Biosite Sangli for providing the CO2 incubator facility during my differentiation study. And last but not least, this work is impossible without the greater sacrifice of these laboratory animals and cell lines. So, thank you. Thank you, ma'am.
Now,
Sense overview, ma'am.
hello, sir, am I audible?
It means, ma'am.
Sir, now the topic is open for discussion. You may ask questions.
If you have any questions, you can please ask.
Hello.
Yes. Hello.
Yeah, this is Vishwanath. Can I talk to the student?
Yeah, see you. Question.
Okay.
You can sit down, otherwise. Bus. Is she
So, congratulations for a nice presentation.
Thank you, sir.
Okay, I went through your thesis,
Yeah, sir.
and I do understand that you do have two cell culture model.
Okay, sir.
One where, in which you have used cytokine for the apoptosis, and you tried two plant extract, one from curcuma longa and another one is from Piper nigris, and it has given a beneficial effect. Am I right?
Okay, sir. Yes,
And the third one, I think you have used another cell line for differentiation, and you have used tinofola for the regeneration of beta cells, in which you have showed secretion of insulin.
Yes, sir.
Okay, I asked one question during the thesis review. So you have used an extraction from both curcuma longa and Piper nigrum as an aqueous extract. Am I right?
Yes, sir.
Why did you do differential extraction with solvents of different polarity? Probably you might have got other active biomolecules.
Yes, sir.
Why did you go for an aqueous extract? I know that reduces a lot of work. I know extraction because you use 10 different plants. Am I right?
Okay, sir. Yes, sir.
Probably the other 10 with different solvents, you might have got some bioactive principle as well. I
Yes, sir.
just wonder like why you have used only the aqueous extract and why you did not go for it. That was the question I think I have also asked in my thesis report.
Sir, we are interested in oral therapy. So the aqueous extract is easy to give orally. So we are interested in aqueous extract, sir. And also
they doesn't show any toxicity.
Okay. Now, this is a general question because when it comes to the type 1 diabetes, I think in your introduction, you say it is principally because of antibodies. Am I right?
Yes,
One is for the insulin, pro-insulin, another one is GAD, and also for the islets. I think now that is a different concept
yes,
because with an antibody, I think it is going to destroy the whole process, in fact.
yes,
And how does your cytokine model mimic that?
yes,
How come your cytokine model? Because that is an antibody-based destruction. And what you tried initially, this is the general question. I know that I think a lot more work has to be done in this particular regard. It is still in the preliminary style. I just wanted to check that your model of type 1 that is using the cytokine storm. I mean, I think probably that is a buzzword in the current pandemic, the cytokine storm. So you used a combination of, I mean, several cytokines for the apoptosis of this your min6 cell line. Am I right?
yes, sir.
Does that mimic the actual type 1 diabetes in humans
No, sir.
so that these two components can be of beneficial? How do you rate that? You got my question?
Yes, sir. Yes, sir.
Okay.
It's difficult to answer that. But
I know because even when it comes to the animal model,
yeah.
see, you have used streptozotocin because that is definitely a massive destruction of beta cell.
Yes, sir.
And now what my point is that, for example, in case of actual type 1 in which it is an immunomodulatory, I just wonder using some of the immunosuppressant currently, I mean, not a strong immunosuppressant like you use it for organ transplantation in a pharmacological level, probably your plant extract might have a better beneficial effect with the. So if you want to address that one, then we need to really have a type 1 diabetes with is an autoimmune disease. I'm just trying to say that probably whatever the active principles you have found out that might be works better in presence of some of the immunomodulators. I would say the immunosuppressant. I know we don't have because it is difficult to have the cell culture model and also the type 1 diabetes, because if you do have a real type 1 diabetes, which is an autoimmune type, probably the other things will be all right. But still, nevertheless, I think you have given some promising results with the Ayurvedic herbal preparations and with the existing model. There is some promising, but I do believe, I'm sure you also believe that still long way to go before we say that, yes, I think we have an active principle, which orally we can give it to the type 1 diabetes. I know that is a serious problem. I think a lot more children in India are affecting the type 1. And we don't know why all of a sudden they develop this autoimmune disorder. But the question is, how do we address that? Second one is that it is difficult to predict the type 1 diabetes because the moment the children will come to the hospital, their antibody titer is already high, GAD is high, insulin, anti-insulin is going to be very high. It's already too late because when you try to measure it, we hardly find any beta cells to even to regenerate the whole thing, in fact. So we need to work out also in case of an early diagnosis, but it is difficult with an early diagnosis because nobody wants to have our child to be tested for type 1 diabetes because so those are all some of these issues. But anyway, good piece of work, and anyway you have published your data in that, so congratulations. And that's all the questions what I have.
See, I just like to say a comment on what you said, Vishnu. See, basically, this is a very preliminary work.
And yeah.
Secondly, type 1 diabetes is a very complex process. It's not just antibodies. It is the CD4 cell, that is the CD8 cells, the macrophages. The whole lot of it is causing, and it is not the same picture in every type 1 diabetic. That is again another problem. You don't have a uniform kind of situation
Correct.
in type 1 diabetic. So what we actually thought was, okay, we may not be able to use immunosuppressants or CD4 specific or CD8 specific. At least the final component which is probably causing the damage is the cytokines. So whether we can at least, have some intervention there. And that is why this whole attempt was made. Like she said in her final slide also, this is just the starting point. It has to
But currently, if I may add, the type 1 diabetes is going to be diagnosed with the GAD antibodies or the insulin antibodies. The moment they claim that, then the branded is type 1. But that should be the majority of type 1 diabetes. What is going to be there? But the other one, I don't know, because at this particular point, it is because of autoantibodies or because of the immune complications. We still don't know. But majority of them, in case of the children, I think currently, I think apart from the regular sugar analysis, they go for GAD antibodies just to see that the islet antibodies is the one, a standard one, in fact, which is they're going to do for the 3. And I don't know about the other ones and how the whole, the standard protocol is going to be there, because it is difficult to argue also with the doctors, because the doctor says, okay, this is the methodology, that's it. And we go by that principle.
We worked in collaboration with Ayurvedic doctor. In fact, she's here. Dr. Chandramukhi Garar. She is actually also treating type 1 diabetic patients with the population. Not exactly this one, but some other components included too. So actually, through such discussions, we had, and with her, we had a possibility that we tried out different plants. In fact, if you see the plants tried out for apoptosis, almost 70% of them show some activity.
Okay.
So, that needs to be worked out extensively, still working out with the different plants, and possibly there is a chance that we may be able to treat type 1 orally.
Okay.
So she has several patients.
Treating type 1 diabetic patients. I have reduced rapid insulin 3 times a day and Lentus for night. I reduce all doses of rapid. I just took them on Lentus for night only. So 3 injections of rapid injections get rid of these patients. This is the first achievements of marketing methods.
Because, ma'am, in your patients' care, in fact, so they all have, diagnosed as type 1 diabetes, and the children don't take insulin and they only take the Ayurvedic preparation?
Okay. What she's saying is they were taking 3 doses of rapid antigen, insulin, and 1 Lantus. So now with her treatment, for about 2 months, 3 months, 3 months, 2 to 3 months, she has been able to totally stop the rapid acting insulin.
Okay.
The child is only on 1 Lantus plus the oral.
Okay. Probably I think this will be another general question. Ma'am, in the current pandemic, especially if any children with coronavirus, their sugar level is going to be uncontrollable. Have you noticed any such type of children with coronavirus, with abnormal, because they were given all kind of insulin to regulate the glucose level.
Easy.
I'm more comfortable.
I will translate it.
This, in this situation, the autoimmune disorders already means there was a संपूर्ण वेगळ्या पद्धतीचे autoimmune disorders आपण न्यूट्रल केलेले आहे. जवळजवळ 10 ते 12 पेशंट मध्ये मी शुगर रिव्हर्सिबल झालेली आहे. काही काळापर्यंत आम्ही त्यांना अँटीडायबेटिक दिलेले आहे, पण नंतर ते टोटल नील होऊन त्यांची औषधं पण बंद केली आहेत सगळीच. आता हे 1.5 वर्षांनी सगळं बघितलेलं आहे, एक्सपिरियन्स केलेलंच आहे.
For her, I should say that she has also developed an antiviral formulation for treatment of corona. She has treated large number of corona patients.
Okay.
Without hospitalization, they have successfully. That also includes me. That also includes me. Okay, I also use the same medicine that she has prescribed, and plenty of them were with diabetes. At least she says 12 patients were with diabetes, whom she has been able to revert back within, say, month or so.
3 months. Longest period is 3 months.
And this formulations is made by yourself or some company makes it?
Sir, I
Personally, she makes the
Oh, okay, sorry.
darling.
Okay.
Lot of hard work.
say thyroiditis, glomerulonephritis, acute pancreatic loss of beta cells, rheumatoid arthritis, SLE, SLN. हे atomic disorders ह्या स्पेस मध्ये खूप वाढले आणि ते रिव्हर्सिबल करायला आम्हाला एकच टेक्निक वापरायला लागते. म्हणजे खूप मल्टिपल थेरपीज नाही लागल्या, एकच सिस्टीमने सगळं सिमेंट सिस्टीमचे रिव्हर्सिबल झालेले. जे आता इथे आपण तुम्ही म्हणता की एकच सायटोकाईन यूज करून तुम्ही किंवा सायटोकाईन स्ट्रॉंग आहे आणि त्याला रिव्हर्सिबल करण्यासाठी व्हेरिएशन पाहिजेच का, तर तसं काही जरूर नाही की तुम्ही जरी आणि मॅडमने जरी 3 जरी ट्रॅक घेतले असतील सायटोकाईन्स मधली, तरी ऑल ओव्हर IS6 आता CRPS मध्ये किंवा ह्या टाईपचे जे प्रोटोकॉल्स तपासले गेले त्याच्यामध्ये स्ट्रॉंग मध्ये सायटोकाईन स्ट्रॉंग मध्ये, ते सगळेच आम्हाला बेसिकली फारच कमी औषधांमध्ये ते रिव्हर्सिबल करतात.
Actually, we had a lot of discussions with her on autoimmune response. And what I was trying to get out from her is she treats various autoimmune diseases like CADR, SLE, and pancreatitis, and so many others. So I was trying to get from her what are the plants she uses for all these. And we tried to, sort of conclude to about some 10 plants from them,
Okay.
which we then took up for study for this beta cell apoptosis.
Good.
They are doing very good work.
Okay.
It needs a lot of validation, not very easy to do. But yes, in terms of patient outcome, yes, they are doing a very fantastic job.
Okay then, I think that should be fine with me.
Anybody else has any questions?
Yeah. Yes, being as a chairman, it is my duty to ask some questions, and preferably I will ask technical questions, sir. As you are one of the referee and you already asked that why only you have used the water extract and she cleared the answer. Second referee is Dr. Vandana Hibrale. Actually, she is the Professor and Head Department of Biochemistry from Babasaheb Ambedkar Marathwada University, and she is reporting very good about the thesis work and all, and very minor comments on your technical errors. So no questions from both the referees. If you would like to ask question from the audience,
Comments or suggestions? Or
yeah. Otherwise, I will ask one questions I have. Actually, I please go to slide number 15. I don't remember that what I want to ask you. If I remember, I will ask. Otherwise, I will just go on.
15 number slide.
15 आणि 21. Because this is it's my duty it's my duty that whatever technical data you are presenting now, most of the students are working on this fluoro and spectrophlorometer. So I don't find any difference between your C and D diagram while taking. Can you please explain this, why these things are happening? C and D diagram.
Yes, ma'am. Detail. In case of 15 nanogram per ml, the early apoptosis is 1.7. And where the
But you did this double concentration, 50 nanogram per ml, and the fourth one is 100 nanogram per ml.
yes,
And that says 19 and 20, and that's why I couldn't find this difference over there. So could you please explain this, why this happens? So can you please say, in spite of using the 100 nanogram per ml, whatever you did, why not 15? Yeah.
because ma'am, we are interested in early apoptosis. Because we are in early apoptosis, when we treat our drug at the stage of early apoptosis, it prohibited all the apoptosis path
Further given.
in early apoptotic stage.
So, maximum.
Okay.
So we are interested in
Just giving early apoptosis we are choosing.
Okay. Go to slide number 21.
21 slide number.
Same question here. I'll just skip this. Cocktail 1 and cocktail 2. I have already asked the question in between. you prefer the things because they are very costly and try to minimize the doses. Okay, so we'll go for the slide number 29. This slide it doesn't give the other things what you have used for the for seeing the effect of CRPN on the cytokine-induced apoptosis, because the description is not mentioned on this particular images. So please tell me the detailing of.
Okay.
आधीच्या स्लाइडला ना जे स्लाइड्स आहेत ना त्याचं काही तू केलेलं आहे ना ब्राईट फक्त इमेज एवढंच लिहिलेलं आहे त्याचं बाकीच्या ज्या गोष्टी असतात ना ते टेक्निकली तुम्हाला थिसिस मध्ये जर समजा आता मी आता वायवाला बघितलंय तू एक्सप्लेन करतेस म्हणून मला कळतं बरोबर, पण त्या स्लाइड्सला असं कसं लिहिलेलं आहे ना तसं त्या स्लाइडला कुठेही लिहिलेलं नाही. मला वाटलं थिसिस मध्ये तू लिहिला असेल तर बाकीच्या निमित्त हे लक्षात ठेवा काय ते तुम्ही दाखवता त्याचं डिटेलिंग कायम राहायचं असतं.
That is only in terms of numbers. 29
स्लाइडला ते एक्सप्लेन केलं तू करशील पण ते डिटेलिंग काहीच नाहीये.
Basically, it is only a picture showing the reduction in the apoptosis झाल्यामुळे हे कमी झाले पण राहिले.
जी वापरलेली आहे ते सगळ्या स्लाइडसाठी एकसारखेच आहे. म्हणजे जे रिसर्च इंटरप्रिटेशन करत असताना ते लक्षात येते. तेवढं तिथे व्हायला पाहिजे. स्लाइड नंबर 36. Okay, so slide number 36 मला हे विचारायचं तर you said that it prevents the percentage prevention of apoptosis, you said. Okay, so how much percentage it is preventing the apoptosis? How much percentage? Percentage म्हणजे किती? 100%, 20%, 10%, 1%?
1.5 fold.
1.5 fold means how what is the percentage?
As compared to
Percentage किती येतं त्याचं? कारण कसं आहे तर तुम्हाला किती पर्सेंटेज समजा 50% अचीव करायचं झालं तर तुमचे डोसेस व्हॅरी होणार त्या पद्धतीने. तर ते असं वेग बोलल्यासारखं मला 2-3 वेळेला वाचलं. Yes, it is inhibiting. Obviously, it is inhibiting. But you should know that the figure, what exactly the percentage inhibition it is giving.
Otherwise, it is not. It is there only you to get.
आता सगळे तू बोललीस, मॅडम बोलल्या, हे बोलले. It is your subject and you understood it. Being as a layman person, I should know that I want to take this as a medicine. Okay. तर त्याचं पर्सेंटेज इनिबिशन जे रिव्हर्सल बॅक दाखवलेलं असते ते मला क्लॅरिटी नाही. ते त्यामुळे तुम्हाला ते कुठं असेल.
आणि दुसरा भाग कसा आहे की हा सेल्युलर लेवल वरती आहे आणि इन्व्हायरमेंटचा वेगळा आहे.
परत वेगळा तो तुम्हाला
what we do is whatever we try, supposing as a thumb rule, what we do is this concentration we have tried out on cell which is working. Okay, so when we do the in vitro experiments in ह्याच्यामध्ये जे करतो आम्ही whole tissue मध्ये वगैरे we use 10 times concentration and when we go to animals we use 100 times.
ते 50% आता फॉर एक्झाम्पल माझं बॉडी वेट इतकं आहे तर ते पर्सेंटेज मध्ये येतं. अदरवाइज द ड्रग म्हणजे लेमॅन साठी इट इज 100% दे आर आस्किंग द क्वेश्चन. ओके, येस, गो टू स्लाइड नंबर 45. Yes, it's good to hear that you are reversing back the beta cells, and it's happy you said that you use this method of staining method. Okay, so how do we confirm? This is the only method.
This is qualitative method.
And we can give the reference. कारण मला ते अपील नाही झालं की एका मेथड मुळे फक्त तुम्ही असं म्हणू शकता की रिव्हर्सल होते. You checked all other activities पण तुम्ही चेक केलेले आहेत. But is this the ideal method used?
That was on staining, actually used for eyelid staining.
Eyelids. Okay, ठीक आहे. Then
बोंडेचं बरंच काम आहे.
ठीक आहे त्याच्यात. म्हणून मला पण मी म्हटलं रेफरन्स मला कुणाचं तेवढं एकच आपण म्हणू शकतो कारण की मला
that has been compounded by checking the PDX, level expression of the
only the carbonic anhydrase is the working marker for this types of study. दुसरी नाही आहे का एक सिंगल पर्सनचं अपरेग्युलेशन आणि डाऊन रेग्युलेशन इट्स नॉट म्हणजे असं मेजर केलं जातं का मला
Ductal cells are major. The biggest this thing is carbonic anhydrase because it produces carbonic.
So it is only
that is the
major
major.
one can use.
Major.
Yeah, major.
आणि insulin producing cells साठी periods मध्ये insulin they don't major markets. आम्हाला so that is how we are doing this selection on these three.
And finally, final conclusion. Yes, conclusion wise it's very good. Again, I am interested in the how much percentage is recovered, percentage wise and body weight. So you should be able to tell this. And if you are saying that this all these four combinations are doing very well and experimentally we have excellently presented, I have no doubt about that. But if this therapy one should want to use in that, and what age or what level I can say it is most effective? This is my first question. And if it is effective, how could you take? How could you popularize? These therapies are available and all these things. So the popularization is equally important because you are presenting your paper publications or everything, and we only the department persons are we are keen to know that yes, these things are to be useful, and preferably we can modify our diet even though we are not diabetic, but we can modify or include these things in our diet at regular routine. But it is to be necessary that whatever finding you have, and if you are damn sure about that, and if you are publishing the paper, so this message should go to the masses. So what attempts you are taking popularization of recipiating your messages, or otherwise you have to develop
That
that
is the first thing.
Yeah.
First thing what
First.
you should do if you want to use this as a therapy.
आणि कुठल्या लेवलला त्यांनी युज करायला पाहिजे हे मला काही हे झालं नाही म्हणजे आपल्याकडे डायबिटीसचे पेशंट सर जसं म्हटलं तसे इन्फन्सी लेवलला पण असतात किंवा चाइल्डहुड लेवलला आहेत आता सध्या त्याचं रिझन काय आपल्याला माहिती नाहीये माझे 2-3 मी थोडं माझ्या डोक्यात आली आहेत मी त्या बऱ्यापैकी मला वाटतंय की हे असावेत रिझन फॉरगेट अबाउट दिस कारण मी मेटाबॉलिझम खूप शिकवतो तुला माहिती आहे कार्बोनेट मेटाबॉलिझम आता इतके वर्ष शिकवल्यामुळे मला ते थोडंसं कळलं पण तुम्हाला जर पॉप्युलराइज करायचं झालं तर मग कुठल्या एज पर्यंत पेशंटनी त्याच्या ह्या गोष्टीला सुरुवात करायची
Ma'am, firstly, we have to check, we have to we have to do clinical trials by using the age groups, different age.
मला
Then
कसं म्हणजे सांगायचंय की डायबिटीस हा डेव्हलप झाल्यावरच लोक येतात तर तो डेव्हलप व्हायच्या आधी तुमचं एक जे आता तुमच्या ग्रुपमध्ये 15 PhD 16 PhD झाल्यानंतर तुमच्या एक थोडंसं लक्षात आलं असेल की आपण ज्या फेज मध्ये आता चाललेलो आहोत तर अर्ली डायग्नोसिस कुठल्या एज पासून लोकांनी सुरुवात करायला पाहिजे आणि समजा 1-2-3 मार्कर जर त्याच्यामध्ये डिटेक्ट होत असतील तर त्याच्यानंतर तुमचं
आपल्याकडे unfortunately like Vishwanath said we don't check for diabetes but abroad they check for that diabetes in the involves they're just generally बाळांमध्ये चेक करतात जे आपल्याकडे केले जातात
तेव्हा त्यांच्याकडे ते कसं आता पर्सेंटेज खूप जास्त आहे ना
they do a lot of tests which we don't do. Second is आपल्याकडे जे टेस्टिंग होतात ज्यांच्याकडे ऑलरेडी एक जेनेटिक म्हणजे नाही तर ते पूर्ण साधारण 8-10 वर्षाचं होईपर्यंत काही कळत नाही then he faints, he falls down, he has all kinds of complications, then they go to the doctor and he realizes that the child is type 1.
So it is mandatory that those who have a genetic background like this, they should go. But what about others? What percentage of these others, those who don't have this genetic background like diabetes and all, they should go at what age?
At the earliest, after the child is born, immediately you have to check.
पण सर मॅडम ती अनप्रेडिक्टेबल आहे आता माझ्याकडे 18 वर्षाची मुलगी आहे तिला आता सुद्धा आहे 1 महिना आधी फक्त आता सुद्धा जायचं त्याच्या उलट एक मुलगा आता खेडेगावात येतोय 10 वर्षाचा पण त्याला दूध खाल्ल्यावर अँटीबॉडी वाढतात आता कुठल्या तरी अजून शोधायचंय त्याच्यावर आजपर्यंत त्याला काळ दूध पिण्यानंतर स्वेलिंग होतं जर त्याला तिकडे तो गोंधळ असेल तर तो डायबिटीस पण लागतो त्याच्या पुढे मी त्याच्यावर काम सुरू केलं बघायचं पण ह्या केसला तर आम्हाला ऑन ऑफ सडनली that the co-vaccine insurance, suddenly insurance, like the sugar level, the insurance passes on that. शुगर म्हणजे आपल्या परिचयातले 60 ते 8 लोक आहेत म्हणजे
Just take that slap to Shubhat and we just say thank you.
व्हिडिओ त्यामुळे मॅडमचं थोडंसं अनप्रेडिक्टेबल मुलांमध्ये पण सांगता येत नाही मॅडममध्ये असं डिवायडर पॅरेंट्स ठेवतात.
So, Professor Vishwanath, thank you so much.
All right, thank you. And congratulations to the candidate.
Yes.
And also for you that you are relieved from all your students.
Yes, finally. I'm through with all my PhD students.
Okay then, good. I think I take leave. Thank you very much for calling me.
So thank you, President. I may say Dr. Hemangee as Dr. Hemangee. Congratulations to both. Okay.
Okay, sure.
Call you later.
Okay then.
Okay. Bye.
Thank you.
Mr. Thank
Thank you, sir.
Thank you, sir. Names. Also, he will come here.
Congratulations.
He's already left. Thank you.
हे कुणाला द्यायचे मॅडमला द्यायचे
कुणाल केलं
ओके मॅडम या सर या त्यांनाही दिल्या त्या दोघांना देऊया मग दोघींना मिळून एक हिला देऊया हेमांगी हा हेमांगी
So, Dr. Budiali, So,
congratulations.
thank you so much.
त्या मॅडमला थोडी त्यांना द्या
We are extremely grateful to you.
Yes.
Not only for the formulation, help in formulation, but the detailed discussions we have had with her and improved our understanding. Thank you so much.
Congratulations.
Thank you, sir. तुमची आवाज नाही. काय बाई रेकॉर्डिंग Thank you, all of you. Yes.